Welcome back, everyone. Let's jump straight into another explainer. Today, we're taking on a massive concept and breaking it down step by step, visually. We're going on a trip right down into the microscopic world of your own body to unlock the exact mechanics of how you're built, how you run, and honestly, how you even survive day to day.
Now, we all know the famous double helix, right? You've heard your whole life that DNA controls everything. It's the core of who you are. But here's the real question, the one that kind of gets glossed over. How does a tiny inanimate molecule actually turn into the sheer complexity of physical biology? Like, how does raw code literally become your muscle, your skin, or the enzymes digesting your breakfast?
Okay, let's dive into this. There's a massive, almost unbelievable gap between that raw genetic code you see on the left and a fully formed, highly complex human being on the right. I mean, it feels like magic, a silent string of chemicals somehow dictating the exact architecture of a living, breathing person. Crazy! But it's not magic. It's a highly ordered, incredibly precise mechanical process called gene expression. Section 1. The Great DNA Myth.
To really grasp this, we've got to bust a major misconception right off the bat. DNA does not directly build your body. Genes don't actually assemble your tissues. Honestly, DNA is practically just a lazy architect. Think about it. A recipe is totally useless unless someone or something actually cooks it, right? DNA needs a worker to get the job done. And this sets up a huge mystery. If DNA is just sitting there holding the instructions, what exactly is doing all the heavy lifting?
Let's break that down in Section 2, the master blueprint metaphor.
Look at how cleanly this maps out. Think of your entire DNA as this massive master recipe book containing every single instruction for your whole body. Now, if you zoom in, a gene is really just one tiny section of that book. It's a single recipe. Say the recipe for a muscle cell or maybe your eye color. But the final product, the actual cooked meal that gets put on the table and does the work, that's a protein. The whole journey of turning that raw DNA recipe into a physical protein meal, that's what scientists mean when they say gene expression.
But here's the thing. The cell doesn't just jump from DNA to protein in one chaotic leap. No way. To protect your precious original blueprint, it uses a really strict two-step master plan. First, it copies the data in a phase called transcription, then it manufactures the product in a phase called translation. So let's look at exactly how this two-phase manufacturing process protects your biology.
Which brings us to Section 3, Phase 1, Transcription.
Let's descend right down into the absolute center of the cell, the nucleus. Notice that heavy, vault-like architecture? Inside, the master DNA stays super protected. And why? Well, you wouldn't take an ancient one-of-a-kind recipe book into a messy, dangerous kitchen where it could get ruined by spilled ingredients or a hot stove, right? Same logic here. If your DNA gets damaged, the whole system breaks down. So instead, you make a disposable copy of the exact recipe you need. That copy phase is transcription. Watch the mechanical precision here. It's amazing. The cell literally unzips the specific section of DNA, just the gene it needs. It exposes those chemical letters, and then an enzyme comes along and captures those exact instructions onto a single disposable strand. We call this new strand mRNA, or messenger RNA. It's called the messenger because its one and only job is to carry this data away from the protected vault. Let's quickly count out these 3 micro steps so we're absolutely clear. Step 1, unzipping the DNA to access that specific gene. Step 2, copying that exposed code onto a messenger strand, the mRNA. And Step 3, that newly formed mRNA strand exits the nucleus. It leaves the vault and heads right out into the cellular fluid, totally ready for the factory floor.
All right, Section 4, Phase 2, Translation.
And this brilliantly illustrates what happens next. Now that we have our disposable instructions, we shift completely out of the nucleus. The mRNA copy arrives at the cellular factory, a structure called the ribosome. Think of the ribosome as this heavy-duty piece of machinery. The mRNA feeds into it almost like ticker tape. Here, outside the vault, the raw genetic code is finally going to get translated into tangible, physical biology.
As the mRNA slides through the ribosome, the machine reads the instructions in small, super precise pieces. And for every piece of code it reads, the ribosome grabs a specific building block called an amino acid. It starts linking these amino acids together, one by one, basically like beads on a string. It reads a code, adds a bead. Reads a code, adds a bead. This string elongates, building the foundational structure of what will soon be our worker. To keep our step-by-step rhythm going, let's map these 3 actions too. Step 1.
The ribosome reads the code from the mRNA ticker tape. Step 2. It translates that code by linking the correct amino acid beads together. Step 3. It outputs a long, linear chain of these connected amino acids. The data has officially become physical matter.
Let's just pause right here to really solidify what we've seen so far. Looking at them side by side, in transcription, we're inside the protected nucleus, turning DNA into mRNA. That's just like copying our master recipe onto a disposable index card. Then, in translation, we move over to the ribosome, turning that mRNA into an amino acid chain. That's like finally cooking the meal from the index card. It's a beautifully elegant, wildly efficient two-part system.
Moving on to Section 5, Shaping the Final Protein.
Now, that string of amino acids, it doesn't stay a string for long. Almost immediately, driven by the chemical properties of the beads themselves, it violently contorts and folds into a highly specific complex 3D shape. It goes straight from a loose chain to a rigid structural block. And here is an absolutely crucial golden rule in biology you have to remember. Shape equals function. The precise 3D shape of this folded structure dictates exactly what job it's going to perform in your body.
And boom, there it is. We finally found our mysterious worker. These folded structures are proteins. You know, while DNA gets all the fame in the magazine covers, proteins are the ones doing the actual physical work. They're the enzymes speeding up chemical reactions so you can digest your food. They're the hormones acting as keys to send signals across your body. They're the physical structural parts that literally lock together to build your muscle tissues. Proteins are, without a doubt, the ultimate workers.
Section 6. Editing the source code. Now, this whole process perfectly explains why modern gene editing, like CRISPR, actually works. Scientists aren't out there trying to manually fix physical proteins or the body directly. That's way too difficult. Instead, using tools like CRISPR, they simply rewrite the original DNA instructions. Because this two-step process is completely automated, by changing the base recipe, the cell automatically generates a new mRNA copy, which then automatically builds a different protein. They're altering the final physical machine at the absolute source code level.
So pulling all the way back, here is the full blueprint in action. DNA hosts the library. Transcription makes the mRNA copy. The ribosome translates it. And out pops the fully folded protein. I really want you to think about this. Right now, as you're sitting here taking this all in, this continuous, non-stop assembly line is happening trillions of times over inside your cells. It is constantly working to physically build, run, and repair you in real time.
We've seen how incredibly perfect this system usually is, but nature isn't always flawless. So I want to leave you with a provocative thought to challenge this new understanding of our delicate system. If altering the DNA intentionally with CRISPR changes the final product, what happens when nature makes an accidental typo in the recipe book?
Well, that typo is a mutation. Changing just a single letter in the original code fundamentally alters the entire 3D physical machine at the end of the line. That one little typo can cause a devastating disease, or it can drive the evolution of an entire species. But we're going to save the incredible mechanics of mutations for our next explainer. Keep learning, stay focused, and thanks for exploring with us.